although I'm going to focus on tau, you'll see that as I say, the intellectual framework is very much in lysosomal storage diseases, I'll make it clear. Just to sort of explain a bit of tauology first, the tangle is the thing that defines Alzheimer's disease, and it's the thing that Alzheimer discovered. And the tangle is made of a polymer of tau protein, and these are the filaments, paired helical filaments. This is a fuzzy paired helical filament, but you can remove all of that. All of the phosphorylated epitopes are simply in the fuzzy coat. You can remove them, but the structural integrity, or more importantly, the proteolytic resistance of the PHF remains. And here we've got a monoclonal antibody which labels the core, and this is an antibody which we developed here in Cambridge, which specifically recognizes a GLU391 truncation site on the tau molecule, which is created by a templated cleavage of the whole tau molecule. And here is the pathology visualized with this antibody. You can see that the tau pathology is extraordinarily intense in Alzheimer's disease. A very important thing, unifying concept in understanding the tau pathology of AD is Braak staging. It begins in the entorhinal cortex and hippocampus and spreads out into isocortex in an exquisitely staged anatomy. We tried to publish a paper to disprove Braak staging, and we actually found that it was right 97% of the time. So we found 3% of the cases where it didn't work. This spread of the tau pathology maps exactly to the spread of neuronal dysfunction as measured by SPECT scan. So Braak staging is excellent. You can see that mini-mental score, which is a very simple cognitive functioning score, maps very well. You can calibrate it very well onto Braak staging once you remove all the vascular pathology. And you can see that the tau aggregation pathology begins very early at Braak stage 2. So Braak stage 2 is kind of mini-mental 30, and then it goes on. And this tau aggregation doesn't turn into tangles until Braak stage 4, and it doesn't turn into dead neurons in neocortex until Braak stage 5. Now, these stages are about 10 to 15 years apart, so this is a very long, slow process, which again comes from Braak's work. He published probably the largest post-mortem series that's been done in Ohm et al. And we did a survival analysis on his data, and it shows the age-specific probability of Braak stage transitions from Braak 0 to 1 or beyond, to 2 or beyond, 3 or beyond, 4 or beyond. You can see that it's simply a function of aging. You can convert this survival function into a prevalence plot. You can see that this is the age-specific decline of Braak 0. Braak 1 is kind of in your mid-40s, 50s. And there seems to be some important transition because pretty well everybody who's Braak 2 goes on to Braak 3. And this is Braak 4 or beyond. And those numbers drop simply because the population here drops. So essentially, once you're at Braak 2, you're on your way. So you can, but it's cognitively trivial, Braak 2. I mean, you wouldn't get a diagnosis of Alzheimer's disease at Braak 2, but yet you're on your way. From this, you can calculate, for example, if you take an average, say, 70-year-old, 75-year-old, and ask, you know, if they have a mini-mental score of less than 25, because you can calculate the age-specific cutoffs for different mini-mental scores, and Braak, you can do a convolution of these two, and you can see that Mrs. Smith, who comes to the clinic age 75, or 70, has a 2/3 probability of being at Braak stage 3 or beyond, irrespective of what other pathology is going on in her brain. Now we know that Braak 3, Braak 4 is about where you become clinically diagnosed as AD. So how does tau aggregate? Well, just a very long story. But in essence, the paired helical filament is made up of a very short fragment of tau, which is precisely this self-replicating fragment. So in essence, the process of tau aggregation is simply a self-propagating protein misfolding that converts normal innocent full-length tau into proteolytically stable toxic aggregates. You can model this in cells. If you have a little bit of this as a seed, turn on full-length tau, it will convert into the truncated form and get massive aggregation inside the cell. This kind of model of autocatalytic propagation was recently confirmed by this study, which showed that if you inject aggregated tau into a normal mouse brain that is expressing human tau, you will simply propagate aggregation, which exactly fits with the cell model. So this is where the lysosomal bit comes in. What seems to be critical in triggering this self-propagating cascade is a seeding or nucleation event. And the field generally concentrates heavily on amyloid and presenilin. But I'm with Anna Maria here. I actually think it's actually driven by a lysosomal storage process, which provides the critical seeding event. Now you can get into this pathway without this, just via tau mutations. And the kind of model that we have in mind is that the real driver is endosomal lysosomal or autophagocytic failure of processing, particularly of mitochondria. Because what you find in lipofuscin in the aging human brain is essentially just 3 mitochondrial proteins, or protein 2 of complex 3, ATP synthase subunit 9, and porin. And it turns out all of these form proteolytically stable complexes with tau. So once this triggers the process, what you have is tau oligomers, the only mechanism of clearance of which is precisely the pathway that begat them. So there is no way out once you get into this pathway. And where does APP and presenilins add? Well, they unfortunately have to be processed through the same pathway. Here is a double labeling showing porin and lysosomes, and we have the other one is core 2 of complex 3, and you can get exactly the same picture in Niemann-Pick and Sanfilippo with no amyloid or presenilin, but you can trigger tau aggregation. Okay, so where does tau aggregation inhibitor therapy fit in? We've constructed a mathematical model which has this loop in it, which really comes from the cell data. And you can ask the question, well, what if you had a drug which blocked on the input side, or what if you had a drug which enhances clearance? This goes directly to Aubrey's fundamental intuition. It's interesting that once you build this model in, you can mathematically model what would happen with a drug that is on the input side. Let's say it's a putative, people would say a kinase inhibitor or some sort of amyloid inhibitor or something. The interesting thing is that all that it will do is produce a transient reduction in aggregate levels, but the age-related accumulation will simply continue. In other words, it will look exactly like a symptomatic therapy, although it's conceptually disease-modifying. The only way to get this curve is to enhance clearance. Now, why the maths works this way is because you can't inhibit perfectly. An inhibition on the input side will simply result in a different kinetic equilibrium, which will re-establish itself at the level of the fundamental block, which I've here shown as simple dams which equal Brach staging. So we developed methylthionynium chloride, MTC, and we showed in lots of models that it inhibits tau aggregation. It works in the cells, it works in vitro, and if you add it to isolated paired helical filaments, which are proteolytically stable, it will disaggregate them and can make them proteolytically susceptible. We made a version of tau forcing it into the endosomal lysosomal processing pathway with a signal sequence, and we got a beautiful tau pathology which respects Brach staging. I won't explain this, but these mice develop a nice cognitive defect and a nice pathology, both of which you can correct with MTC treatment. There's another tau model here, which is a double mutant, one of the so-called FDP models, and it produces a very florid tau pathology, and you can reduce the pathology and you can improve behavior with MTC treatment.